Now I am going to discuss about feeding in amoeba. All of we know amoeba is microscopic animal. It is also microphagous animal. Because micro means microscopic. Microphagous. Amoeba is microphagous animal. Micro means microscopic and phagous means who eats. Takes microscopic plants and animals as their food. Amoeba is a microscopic animal, simple most animals. Still, its feeding is very complicated. Amoeba takes its food in various ways. Amoeba cannot procure its food from simple substances, so it depends on solid organic material for their food. The process by which amoeba takes its solid food, it is known as zootrophic or holozoic nutrition. Amoeba ingests algal cells, algal filaments, diatoms, bacteria, ciliate protozoa, rotifera, small nematodes as their food. Ciliate and flagellate protozoa are their choice of food, are their favorite food. A very surprising fact that amoeba can identify between organic and inorganic substances. If carbon particles with mixed up with their food, it will ingest only organic food. And that carbon particles, they can discard the carbon particles. They will only take organic food. Amoeba can intake their food by any part of their body, but generally they will use anterior pseudopodia for this purpose. So, nutrition in amoeba consumes whole animals and plants smaller than their own size as food. Their feeding mechanism is holozoic or zootrophic, and this method is known as heterotrophic. It has 4 steps: ingestion, digestion, assimilation and egestion. First point is ingestion of food. Amoeba ingests food by means of its pseudopodia and forms a food cup. This one is very much important. Food cup formation in amoeba, first step of their ingestion. Food cup formed in the cytoplasm. When it comes across a food particle, it encircles the food particle, its pseudopodia, without touching the food. Remember this point, without touching the food, and fix the food on the substratum. Only the pseudopodial tips are fused so that the food is automatically transported into the cytoplasm. Following method of ingestions are employed. The first one is circumvallation. So what is circumvallation? This is a food particle. This is amoeba. Food particle in front of it. What will be its next step? Food cup is formed without touching the food. Next, this will be joined. This is circumvallation type of food intake. Okay. This one is called food cup. Active food. This one is known as food vacuole. This is circumvallation process type of food ingestion by amoeba. This method of ingestion is applied when it is a living prey, moving living prey. Circumvallation is the process of capturing living prey living prey. When the prey is living, okay, circumfluence, and this method is applied when the food is immobile, not active, immobile. And in this method, food cup is also first of all, what happens? First of all, pseudopodia is going to touch the food. Circumvallation is without touching the food, and circumfluence, as it is immobile food. Immobile. First of all, the protruding pseudopodia, it is going to touch this food, and next the food cup is formed. Then the food is pinned down. To the substrate. What happens? A pseudopodium is extended below the food to complete the food cup and food, and along with a drop of water enclosed in a food vacuole. And by repeating this process several times, they can ingest a long filament of algae. See here, the food particle is immobile and pseudopodia. Successive position of pseudopodium. These are all pseudopodium. Successive pseudopodium. This is food, immobile food. And this is the body of amoeba. And when it ingests a long filamentous, say this is long filamentous algae. Long filamentous algae. Long filamentous algae. This is long filamentous algae. Long filamentous algae. This is body of amoeba. This is body of amoeba. So this is circumfluence process. Next process, import. When the amoeba comes in contact with the food sinks passively in the endoplasm. Then a food vacuole is formed within which the food is placed. Passive method of taking food is called import. Next, invagination. Next type of ingestion of food is invagination. In means interior, vagination means folded into interior, folded interior. Amoeba comes in contact with the food, then
adheres to it. The ectoplasm along with the food is invaginated as a tube in the endoplasm, and the food particle is sucked in. The plasma lemma disappears and the ectoplasm changes into endoplasm. A food vacuole is formed and the tube disappears.
The next is digestion in amoeba. Food is absorbed, and what about digestion? The next process. And after ingestion, the food is placed within a vacuole along with some water or other liquid, and the digestion starts within the food vacuole, also ends there. This type of digestion is known as intracytoplasmic digestion, and this intracytoplasmic digestion is towards the first step in the evolution of digestive system. First, the food vacuole become larger as some of liquid water is also absorbed. Gradually what happens? Gradually the liquid diffuses out into the cytoplasm and the food vacuole diminishes in size. It has been experimentally verified that this time the pH of the food vacuole is become acidic. The acidic nature is very mild, not very strong, is near about pH 5.6. And after an hour, acidic environment become neutral and finally become slightly alkaline, having a pH of 7.1 to 7.3. It is seen that osmotic pressure of the food vacuole is now increases and the liquid enters the food vacuole from the cytoplasm again, and that's why it's become alkaline. In alkaline medium, the food is broken into pieces and its mass get reduced, and digestion continues within the food vacuole till undigested material are left within the vacuole or in secretory stage. This is food. These are digestive products, and this one is tongue shaped diverticuli. And what is this condition? This is food vacuole in secretory stage. These are digestive products. These are budding vesicles. Budding vesicles. These are undigested product. Undigested fragments. These are undigested fragments. This is food vacuole in absorptive phase. Okay, food vacuole in absorptive phase, and this is tongue-like diverticulum. Tongue-like diverticulum representing absorptive phenomenon. And another is this one is budding vesicle. Budding vesicle, and this one is food vacuole in membrane. Digestion process. Digestion process. All we have discussed about alkaline acidic phase and alkaline phase, and these two phases together constitute digestive vacuole. And there are some enzymes, and how enzyme act and their actions are as follows. First of all, protease enzyme. Protease enzyme has cathepsin I converts protein into peptides, and next one is lipase. Lipase converts fat into fatty acid and glycerol, and amylase converts carbohydrate into glucose. Digestion, the food vacuole is broken into small vacuoles, and with the streaming movement of protoplasm, the digestive food is distributed. Digestive food is distributed within the cytoplasm by diffusion. The digested food assimilated and some are stored as glycogen, lipid, etc. Undigested wastes are expelled out body by exocytosis. The entire process of digestion is intracellular. What happens? Proteases break down proteins into amino acids. Amylase breaks down starch into glucose, and lipase breaks down fat into fatty acid and glycerol. Amino acids are using building in cytoplasm, where glucose and fatty acids giving energy for its work. And various salt solution, protein solution outer the plasma are taken in the endoplasm by pinocytosis. Then comes to assimilation of food. Next point, assimilation. Assimilation of food. The cytoplasm surrounding the food vacuole give rise to many small processes. These processes may increase the secretory area of cytoplasm. Soon, many lysosomes appears and the digestion process begins. The digested material are temporarily stored in places surrounding the food vacuole. Unlike outgrowth, as I have already drawn here, emerges from the inner wall of the vacuole, and is believed that this tongue-like process is responsible for absorption. Gradually and slowly, small bulbs filled with digestive material appear on the wall of food vacuole. Finally, they become detached from the food and undergo cytosis within the endoplasm. The digested food is absorbed and the buds disappear. Now, last point, egestion. Egestion. This is fecal plate. Fecal pellet forming. This is the end point of amoeba body. Fecal pellet forming. Then what happens? Fecal pellet emerges out, expelled out. Fecal pellet inside, then some part is emerges out, and finally it is being separated and the fecal pellet expelled out. Fecal pellet expelled out. So this is the step by step egestion process, fecal pellet forming and expelled out from the body of amoeba. What is egestion? Undigested wastes are expelled out from any side, any part of the body as fecal pellets. Generally, the pellicle of the posterior part bursts and the fecal pellets are expelled, and immediately a new pellicle is formed to stop the outflow of the endoplasm. This is in a nutshell the feeding and digestion of amoeba in microphagy habit.